All right. Thank you for having me. I'm actually very excited to speak at this symposium. I'm a computational researcher at UC San Diego. Chromosomal rearrangements are seen quite often in constitutional disorders. I don't think that's news to this audience. There's, for example, deletions. In this case, a deletion at chromosome 4 can cause this syndrome. But sometimes constitutional disorders can also be mediated by balanced translocations and so on, where without the copy number change, sometimes it can become very difficult with standard of care methods like chromosomal microarrays to be able to detect those. And what we would want to do, of course, is to try and give a more complete picture. So as computational scientists, it was kind of intriguing to us that very old technologies like cytogenetic karyotyping, which existed back in the 70s, already could pick out changes in the chromosome rearrangements, including sometimes balanced translocations. Maybe the resolution wasn't as good, but you could see that there was a very clean picture, as you can see on the slide, of what the chromosomal content of the donor is. So we wanted to, try and build this karyotype digitally. And of course, when you think about karyotyping cytogenetically, you have a picture, and that's the karyotype. But for a digital karyotype, we at least need to give some definition. And the way we think about a karyotype is just that there are many segments from the donor genome, and those segments are represented using reference chromosomal coordinates. And then the karyotype itself is a chain of these genomic segments. So in this case, every chain would represent a haploid chromosome, ideally with 2 telomeres, a centromere. And then by just looking at them, you would be able to tell what rearrangements have occurred. And of course, we would also like to describe the karyotype in a standard form like ISCN. We'd like to identify disrupted genes and maybe use databases to predict what the phenotype might be due to those changes. So very quickly, just to cut right to the chase, you can see an example of an ideogram that's generated automatically by our tool, which has utilized only Bionano Genomics optical maps. So with the optical maps as input, we can get the karyotype, including the text description. And then you might say, well, is it still relevant? We are already pretty good at identifying structural variants and other rearrangements, and that's what we really want. So let's just take a few examples to say how we can add value to that product. So we started with Bionano Access, which is a standard software for people using the Bionano Genomics data, and this is a typical output. On the left side, you can see all the chromosomes arrayed in a circle, and rearrangements are shown by edges inside the circle, and you can see that there's a translocation, and you can sort of dig deep in, and if you can see my mouse, you can see that there are both interchromosomal and intrachromosomal rearrangements, but the overall picture still remains complex, right? Some trained cytogeneticist or researcher has to make sense of all this data. In contrast, if we run this data through OMKar, we would get a clean ideogram of the rearranged chromosomes, right? And a text description of what happened. And perhaps this might seem still to be complicated, but if we just identify the centromeres and the telomeres, you can see that there's a very clean picture that emerges. So these are the centromeric regions, and you can imagine that these are the P arms. And I've sort of drawn this in an inverted fashion just to show that if you think about this as the wild type, in the derivative chromosome, there's a clear balanced translocation that moves, that rearranges the chromosome 9 P arm with the chromosome 22 Q arm. And then moreover, you can see that there is a segment in the derivative chromosome 22 that has inverted and duplicated. And all that is shown both in the figure and also, just very quickly in the Bionano output with chromosomal coordinates. So hopefully that gives a sense of where we are going with the OMKar. Here's one more example, also very complex. You can see the Bionano Access view, very nicely detected all the rearrangements, but then the OMKar analysis can output an improved understanding of what's happening. Here I'm also showing the ISCN-like language that we automatically produce, including plain English description of what might be happening in this chromosome. So it hopefully, even though a trained person has to still look at the data, it can give like an easier way of interpreting the complex rearrangements that are happening. Right? So in this talk, or whatever remains of this short talk, we will just quickly describe how OMKar works, and I'll point you to some results. And I hope that you can look at a more detailed description in the paper that was published. I should also mention two very talented graduate students in my lab, Siavash Raisi and Zhao Yangjia, who did most of the work that I'm talking about today. And we had a very nice collaboration with some Bionano genomics people, as you can see on the author list. All right, so what it does is it starts actually with standardized copy number and SV calls. We decided not to reinvent the wheel. We use the access pipeline from Bionano, but people can make their own SV and CNV calls and put it in a standard format, and OMKar can take that. And then the next step would be to construct the breakpoint graph and smooth edge multiplicities, and I'll show those in the next few slides, right? So if you think about the actual karyotype, we can encode the features of the karyotype on the left using a graph. Every segment in the karyotype can be thought of as an edge in the graph with a head and tail node. So you have these A, B, C, and D, 3 segments. And then you have edges connecting the segments that might be reference edges, connecting adjacent segments from the reference, or they may be SV segments which connect segments that are not together on the reference. For example, B is connected to D, but are together in the donor, right? So that's what we mean by a breakpoint graph. And then we can give copy numbers to all the edges and so on. But the main take home is that the breakpoint graph, even though it encodes the karyotype, is not, you don't need the karyotype to reconstruct it. We can actually construct the breakpoint directly from the CNV and SV data. And so here we invert the problem. We start with the breakpoint graph, and we decide to say what the chromosome is. So we take the breakpoint graph, and the next thing we have to do is to smooth the edges. And the way you can think about it is that there's some flow in these edges. These are all the structural variants and all the reference edges feeding into this. And the copy number here should be more or less the sum of all these copy numbers, right? And maybe there's some slack that you tolerate. And so we get this equation, this constraint that must be satisfied by applying both the slack numbers and the copy number variable that we are seeing out here, right? So an integer linear programming optimization automatically produces the slack, and it penalizes for the difference between the slack and the observed copy numbers. It also penalizes for the slack itself, so you want it to be as small as possible. It penalizes for any change in orientation that might still satisfy these copy numbers. And then finally, we want to make an Eulerian graph, which I'll explain in the next slide, and it penalizes if there are odd degree vertices. So that's how we run this, and the output of this is just a smoothing of the copy number edges. And finally, we have this graph with smooth edges, and then we want to find a traversal of this graph that uses every edge exactly once. And that traversal, as you'll see on the next slide, it helps us determine the karyotype. So here we have a graph, like the karyotype graph that I showed previously. You could have a traversal. It goes like this, and you can see there are 2 odd degree vertices, so it kind of has to stop here. But sometimes we add a dummy edge and we complete the circuit. Once we have this Eulerian circuit, we can break off at the telomeric regions here, and if we have the dummy edge over here. And now you can see that we have these two paths, each of which represents a karyotype, right? So hopefully that gives a sense of how the system works. And then we do a little bit of comparison against the reference to produce a plain language ISCN-like description, and also to see if there are any interrupted genes, right? And so once more, this is what the final report looks like. We have the stylized ideogram. We have the ISCN plus the plain language description of all the rearrangements. If there are any disrupted genes, they're shown in a table. And then there are also, these are, fully supported, for which we have the full explanation. We also have some reports on partially supported, and the very raw output is shown at the bottom. So people can dig down as they might like. And we're constantly working to refine the output. So we'd love to have comments. Okay. Just very quickly, how we evaluated, how we did on this karyotype. We decided to do both simulations, which somehow are not widely utilized in existing literature, but I think tell us a lot about how the karyotyping is working, because we know what the karyotype is. And then we took data, 350 samples or so, collected from different sources, and validated using other standard of care methods. And here we don't know exactly what the karyotype is, but we have some idea. So so it's real data, but we have less knowledge of what the ground truth is. So on the simulations, this is a complex chart, but it shows how we perform. There can be very complex rearrangements, and that's sort of illustrated by this cluster complexity, from very simple to very complex. And the way we measure similarity is through a Jaccard coefficient, which is just intersection of the prediction versus the ground truth divided by the union. So if it's 1, then we are perfect. And if it's less than 1, then we are less than perfect, right? So we'd like to be in this row. And you can see that despite complexity of clusters, we are largely in this, but there are a few cases where we don't always make the right answer. And this is true both for structural variants and also concordance of copy number changes in our karyotype. So these copy numbers are after smoothing. On the real data, of course, like I said, we don't have full karyotypes. But we looked at all the SVs that were predicted by any technology and just took the union of those. And you can see in this table out here that OMKar predicts more SVs than any other method, but also it has a higher accuracy in the ones that it does predict. And you can see all these numbers out here for comparison. And here you can see it on a sample by sample basis. Every column here is a sample. If it's green, that means that these methods predicted correctly. When you see a red, you can see that there was an incorrect prediction or a misprediction. And you can see that by and large, OMKar is making the right predictions everywhere. All right. So hopefully that gives some confidence that we are on the right track. And these SV-like analyses cannot say how good the full karyotype is. So in addition to being correct, we also like to say that we have a simple and more easily interpretable output. Okay, so a couple more examples just to showcase our method. We took some examples where the previous standard of care wasn't very clear, but you can see that there were some, there was some dysmorphia that was observed. And this is the OMKar output on the same sample. You can see there's many complex rearrangements. We can see that there are translocations, transpositions within the chromosome, deletion, inversion. And so we get a complete picture of what's happening in these rearranged chromosomes. But I'd like to focus on the deletion of these two genes. If you sort of look through the literature, you can see that variance in this WYF3 gene is actually known to cause some neurodevelopmental disorders and microcephaly and so on. And so our analysis, even though it's not a prediction, definitely points in the same direction. Right. Here's another example. Again, just to say that the clinical indications were not very, very clear. And one more time, the OMKar output gives a very nice and complete picture of the rearrangements. And we find that a transposition, which doesn't change the content, but it rearranges chromosomal segments, interrupts a gene. And this gene is known in the literature to have some neurodevelopmental disorders associated with it. So again, the karyotyping produced by OMKar allows you to get a better sense of what's happening in terms of the phenotype. All right, so I'm actually done with my presentation more or less, but I want to end with a few thoughts. So is the karyotyping problem solved? And no, it's not. There are still, ideally you want a complete picture down to the base pair level, which we don't have the technologies to do it. But also we are still struggling in complex regions. And also there are some somatic diseases, cancer being one example, where the rearrangements can be very, very complex. And we are doing some other work, including using bionanogenomics data. And here's one example I want to show. So you can see that if you look at the copy number, most of this P arm of chromosome 7 is completely normal with copy number 2. It's on a log scale here. But you can see that there's this small region with nearly 100 copies. So there's no way that our karyotyping is going to explain these 100 copies. It turns out in this case, these extra copies are not even on the chromosome. When you look at it under a microscope, you can see that these are circular extra chromosomal DNA particles. And we have been doing a lot of work on ecDNA. But one of the things that we did is to take the bionanogenomics data and reconstruct the exact content of the circle. So with respect to karyotyping, what we can think is that when we have these complex rearrangements, we can first remove all these circles and then try to do the karyotyping on the rest of the genome. Here's another very complex example, again, of very high focal amplification. Same copy number otherwise, but this, this region is in hundreds of copies. But this time it's not extra chromosomal. These are all copies rearranged on the same chromosome. And it turns out in this case, it's an operation, it's a collection of breakage fusion bridge cycles where the segment sort of rearranges on itself through inverted duplications and makes very huge copy numbers, but they are chromosomal. And once again, we have methods that can take bionanogenomics data and optical genome map data and identify breakage fusion bridge structures completely. So, piece by piece, we are trying to get at some of the complexities that are involved in karyotyping, and hopefully we will be able to combine all these into a full karyotype for many complex rearrangements. All right, so I want to conclude. I hope that you can agree that OMKar allows somewhat accurate and automated karyotyping directly from optical genome mapping data. And in this way, it can kind of bridge the gap between traditional cytogenetics, which is not very high resolution, and sequencing, which is very, very high resolution. It kind of provides an intermediate picture. And of course, it does have limitations, and we are working on overcoming them, both through hopeful improvements in technology, but also in our computational methods. And we'd like to sort of incorporate the rearrangements that we have detected in cancer in this. Okay, so lots of great people to acknowledge. One thing I didn't mention is we had a very nice collaboration with the medical group in Vienna, especially with Paul Dremsek and many other collaborators. And also the Bionano team was very helpful in helping us understand their data better. And so they are all co-authors on the paper that we have published. Finally, our cancer work is supported by a Cancer Grant Challenge grant from CRUK and the NCI, and our other rearrangement work is supported by NIH, and I'd like to acknowledge all of them. Okay, so I think I can stop here. Thank you very much for listening.